Welcome to my channel College Botany Online Free Learning. I am Dr. Yuvraj Kengar, working as Assistant Professor in Botany at Srimati Kusumtai Rajaram Bapu Patil Kanya Mahavidyalaya Islampur, Maharashtra, India. This video is made for online teaching to my student studying in B.Sc Part 3rd, Semester 5th, Botany. Today we are going to learning the syllabi that is just implanted in this new academic year. Under CBCS pattern, choice based credit system pattern, the syllabus to be implemented from June 2020. It has 4 papers in semester 5 and 4 papers in semester 6. First of all, we will learn the nature of question paper and scheme of marking. Each question paper has 3 questions. The first question is multiple choice question. 8 questions are there. Each carry 1 mark. Sum total comes 8 marks for the first question, multiple choice questions. Question number 2. Attempt any 2 of following. This is essay type question or broad answer question. They will give 3 questions. Out of that you have to attempt 2 questions. Each question carry 8 mark. Sum total comes 16 marks. And last question, that is question 3rd, write short notes, any 4. They will give us the 6 short notes. Out of that you have to attain the 4 short notes. So sum total of the all questions lies for 40 marks. The paper numbers as per semester 5th, the paper number 9, its code is DSE E25. The title of this paper is Genetics and Plant Breeding, will deal by Magdum sir. Paper number 10, its code DSE E26, Microbiology, Plant Pathology and Mushroom Culture Technology, will deal by DT Patil sir. Paper number 11, code DSC E27, the title is Cytology and Research Technique in Biology. It will deal by me. And paper number 12, DSC E28, title Horticulture and Gardening, will deal by Mujaur sir. So the papers for the semester 6 we will discuss in the next semester. So I will deal the paper number 11, DSCE 27. Its title is Cytology and Research Technique in the Biology. Total marks for this paper 40 marks and 10 marks will for internal. You are looking here the syllabi of this paper. 4 units are there in the syllabi. The unit number 1 is the cell as unit of life. Unit 2nd, cell organelles. Unit 3rd, subcellular structures and cell membrane. And unit 4, research technique in biology. So let's glimpse on the syllabi. The unit 1.1, the cell theory, prokaryotic and eukaryotic cell. 1.2, cell cycle and apoptosis. 1.3, cell division, mitosis and meiosis with their significance. In detail discussion we will made after some minutes. Unit 2nd is for the cell organelles. We will learn the nucleus, its ultrastructure, nuclear envelope, nuclear pore complex, DNA packaging in the eukaryotes. In detail we will learn the mitochondria, ultrastructure, semi-autonomous body and their role, chloroplast, ultrastructure, semi-autonomous body and role, and we will learn the ribosome present in prokaryotic cell and eukaryotic cell. The unit 3rd is for subcellular structures and cell membrane. 3.1, endoplasmic reticulum, Golgi body and lysosome, their structure and role we will learn. 3.2, peroxisome and glyoxysome, the structure and role we will learn. 3.3, cell membrane, structure, and in detail we will learn the fluid mosaic model and their role. 3.4, type of membranes as per permeability. The last unit, unit 4, research techniques in the biology. 4.1, the principle of microscopy, light, fluorescence and electron microscopy, scanning electron microscopy. 4.2, calorimetry, spectrophotometry, micrometry, photomicrography. 4.3, intellectual property right, IPR, concept and importance. And 4.4, the patent, their objective, procedure and working. So this is the syllabi of paper 11, cytology and research technique in the biology. So just to make a small glimpse of every unit, in the unit first we will learn the cell as a unit of life. We are people knowing that the cell is basic structural and functional unit of life. In this unit we will learn the cell theory postulate made by Schwann and Sheldon. We knowing that there are two types of cells, prokaryotic cell and eukaryotic cell. These are based on the nucleus condition. The nucleus is in primitive condition or in nucleoid, that cell is called prokaryotic cell, whereas the nucleus is advanced, means it has nuclear membrane, nucleopore complex, then it has nucleoplasm, nucleolus, chromosomes. That type of cell is called eukaryotic cell. The bacteria like organisms exhibit the prokaryotic nature of cell, whereas the plant and animal exhibit the eukaryotic cell. Here in this unit we will learn the cell cycle and apoptosis. The cell showing The cyclic changes, that is called as cell cycle. Cell undergoing 4 type of phases, that are G1, S, G2 and M. M means cell division phase, and apoptosis means sometime cell suddenly decide for the death, that is called as apoptosis. Two type of cell division you all people knowing, mitosis and meiosis. Their phases, their stages and their significance we will learn in detail in this unit, unit number 1, cell as a unit of life. The unit number 2, the cell organelles. The prokaryotic cell generally not exhibit the cell organelles, but eukaryotic cell, either plant cell or animal cell, showing the cell organelles like nucleus, mitochondria, chloroplast, ribosome. It has endoplasmic reticulum, Golgi complex. Then it has microbodies like a lysosome, glyoxysome, peroxisomes, and the eukaryotic cell has a cell membrane. So the cell organelles we will learn here in detail. The nucleus is the active center of a cell. In detail we will learn the ultrastructure, then nuclear envelope, type of nuclear envelope, then nuclear pore complex, how the DNA get packaging into the chromosome in a eukaryotic cell. In detail we will learn. We all people knowing the mitochondria is called as powerhouse of cell. Its structure we will learn. Similarly, we will learn the chloroplast. Chloroplasts are present only in the plants with which the plant perform the photosynthesis. Its ultrastructure we will learn. Both mitochondria and chloroplast has its own genome, DNA. That's why some proteins they synthesize and some protein they imported outside the cytoplasm, means from the cytoplasm. That's why they are called as semi-autonomous body. Some protein they synthesize themselves and some proteins imported from the cytoplasm and nucleoplasm. That's why they are called as a semi-autonomous body. Their role we will learn in detail. The last point of this unit, ribosome. Prokaryotic exhibit 70S type of ribosome and eukaryotic cell exhibit 80S type of ribosome. Ribosome is very important in protein synthesis. That's why the ribosome is called as protein machinery. In the next unit we will learn the subcellular structures like endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes, glyoxysomes, and cell membranes. There are several models of cell membrane. Unit membrane model, then fluid mosaic model. Unit membrane model proposed by the Robertson. The fluid mosaic model proposed by the Singer and Nicholson. Their role we will learn here. And last point, the type of membrane as per the permeability, that also we will learn in this unit. The unit 4, research technique in biology. The 4.1 is for the microscopy. Here in this unit we will learn the light microscope, fluorescence microscope and electron microscope. Electron microscope has two types, scanning electron microscope and transmission emission electron microscope. So SEM and TEM, these are the two types of electron microscope. Out of that, the scanning electron microscope we will learn as per the syllabi. Then the principle behind the colorimetry, then spectrophotometry, micrometry, photomicrography, that also we will learn in this unit. 2 points newly introduced in the syllabi, that is intellectual property right, what is the IPR, concept and importance also we will learn here, and last that is patents, its objective, procedure and working. So marking scheme is not adhered with the units, however every unit may have one long question, two short notes and compulsory 2 MCQs. Therefore, the weightage of every unit may be near about 14, 12 to 14. So in detail all these things we will learn in online as well as in classroom lectures. So I have just discussed the syllabi of paper number 11, cytology and techniques in the cell biology. Thank you very much. You have to subscribe this channel because all video will be put on this channel, College Botany online free learning. Thank you very much.